So all of this research has been conducted by Stephanie Talmadge. I won't call her Dr. Talmadge yet, but she'll be defending her PhD on these results in only a couple of weeks. And she's done a fabulous job at pioneering all of this work right here in Southampton. Published this paper in the Proceedings of the National Academy of Sciences just last fall. And she looked at how changing levels of CO2 affect these bivalve larvae and considered not just what's going to happen in the future, but she also considered what happens if you look at the CO2 levels that we used to have on this planet. And what's shown here is her results for bay scallops, considering 4 levels of carbon dioxide. The levels we used to have, the levels we have today, and the levels we're projecting forward. And this is how long what percentage of the larvae survive over a 5-week period. And the results are clear that even the change that's occurred between what we had in say 1750 and today, that change in CO2 is enough to significantly depress the survival of larval shellfish. She did some great electron microscopy work, and that's shown here. These look like regular scallops, but they're really microscopic grains of sand. But you can see when you put them under the low levels of CO2, they look like scallops. But as you increase those levels, they start to lose that shape. These are hard clams, Mercenaria mercenaria. The graph is different, but it looks very similar. Same trend. That is, increasing levels of CO2 are decreasing the survival, which leads us to state that the changes in CO2 that have occurred in just the past 150 years or so are enough to significantly depress the survival of clams and scallops. She conducted an experiment to see, well, what's the shortest period of time we can expose these scallops to see that they survive. And looking at this result today, Stephanie, and I'm sorry, this experiment was a failure because we couldn't find the minimum duration, because even a 4-day exposure to these high levels of CO2 resulted in a significant decline in the survival of these scallops. And certainly you can see the longer they're exposed to the CO2, the smaller their survival rate is. But even 4 days gives you a decline that's significantly greater than what you had if they had pure ambient levels of CO2. Following up on some of that electron microscopy work, I just want to show the hard clam electron microscopy shown here. Similar pattern with regards to size and shape. She was able to cut those shellfish right down the valve there and look at the inside of them, and you can see these shellfish look large, robust, and thick shells under the lower levels of CO2, and as those levels get higher and higher, they become very thin, very frail, and very brittle. And then specifically looking at the hinge of these organisms, this area right here, the hinge is lock and key under ambient or low levels of CO2, but again, you increase those levels and that hinge starts to disintegrate. So clearly we're affecting not just their survival, but the integrity of these organisms as well. Stephanie's also does some work looking at what happens when you put these organisms out in the field after that larval stage. So that is, we'll just give them 3 weeks of high CO2 or low CO2 and then see how they do. And on the upside, the ones at the higher level of CO2, although many of them perished, the ones that survived caught up to the ambient level of CO2. But surprisingly, the ones at the CO2 levels we used to have continue to do much better, well, 20% better, than the individuals that were at ambient level of CO2. And this data is only up to March 1st, but I know that she went out this week, and the pattern still holds this week. And that is, even after 10 months, they've only had 3 weeks of exposure, but that legacy of that larval effect is still present 10 months later, and they're still doing better, even though the exposure was only during the larval stage. So we know our shell fisheries have declined here in New York, and we know that the reasons for these declines have been harmful algal blooms, over-harvesting, but I think we can't deny the potential impacts of acidified coastal waters that may be contributing to some of these declines as well. Now, we assume so far that all these impacts would just be on calcifying organisms, but more recently we've been looking at non-calcifying organisms. And Dr. Hannes Baumann is here. Raise your hand, Hannes. And we've been collaborating on some experiments along with Stephanie, looking at the effects of CO2 on fish, specifically Menidia berylina, the inland silverside. It's been assumed by scientists up until now that fish will be immune to high levels of CO2. These are large organisms, and people have done lots of research for many years on these organisms, that large fish, when you expose them to high levels of CO2, do just fine. But we specifically have been looking at larval fish, so that is, and the eggs. So that is, what about these very small fish and these egg stages that people have not looked at yet? And when we roll back the life history and look at this very first stage, we find they are sensitive to CO2 as well. Here's the percent survival of those Menidia eggs and larvae exposed to different levels of CO2. And you can see that even small increases in CO2 significantly depress the survival of these fish, and also significantly decrease the size of these individuals. And we've done some work looking at them under the microscope. Lucas Merlo is here somewhere and took these pictures. And he pointed this out. He said, well, these fish here are pinned straight, but at the high CO2 levels, they start looking a little strange. And in fact, there is a higher incidence of deformed morphology of the fish exposed to high levels of CO2. Oops. Now, I've been talking about CO2 as something that's in the future in the open ocean. But there's reasons to believe that coastal zones are now threatened by acidic water and high levels of CO2. Here's a data set from Chesapeake Bay. Only the last 25 years, the levels of pH, or pH levels, are declining in Chesapeake Bay. And this is up in Maine, showing discharge from a fresh water source, showing that by having more fresh water discharge into a coastal zone, you can depress the saturation state of calcium carbonate, which essentially is giving you low pH, high CO2. So we know that there's sources, global sources of CO2 that are affecting us, but we also know there are local sources, and some of those, again, link back to nitrogen. Because if you have higher loads of nitrogen, you're going to be increasing algal biomass, and there's a seasonality to this as well. This algal biomass will increase through the spring, maybe in the summer, but eventually that degrades and it goes away. And when that happens, you're driving up the CO2 and driving down the pH. And it may be that sewage treatment plants and power plants may be another source going forward. So, as I said, our fisheries are on the ropes. And it's not just our fisheries, but also our eelgrass. I've talked to you about the problems with regards to nitrogen and acidification. I'm going to now turn it over to Brad Peterson, and he's going to tell us what we can do to fix this problem.
Well, Chris took 45 minutes to set the stage, and he told me that I have 7 minutes to try to explain how to take care of everything. After listening to Chris for the last 45 minutes, you're sitting there and you're wondering to yourself, what do you do? You saw the pictures of the ostrich with his head in the sand. What do we do? Well, unlike the animals, we have a will, and we can choose to respond. And so what I'm going to now talk about are possible ways that we might respond in the face of what's going on locally, and I'm going to fixate my response on Shinnecock Bay. So this is just to orient ourselves to Shinnecock. And Chris and I have been spending the last 8 years trying to understand the mechanisms for the things that are going on there that are impacting seagrass and the survival and growth of seagrasses. And we've learned many things. One thing we've learned is that from the inlet, as we move towards Quantuck, there is a decreasing light availability that's driven by algal biomass, so poorer water quality. And this is just a plot looking as we go away from the inlet, our productivity and survival of the seagrass drops. And at the same time, the chlorophyll levels go up. And not only do chlorophyll levels go up, but as Chris talked about, the size of that chlorophyll drops. And that has a really important impact on the things that eat that phytoplankton. So we could think of it as a progression of nutrient enrichment from the inlet further towards Quantuck. Right by the inlet, we have low nutrient levels in the sediment, so the grass is actually inhibited But as we move away, we start to see this enrichment of nitrogen leading to epiphytic growth or algal growth, and then being dominated by phytoplankton that's totally shaded the bottom and allowed no benthic production to occur. So what we want to do is we want to say, how can we go in this direction? How do we move against this nitrogen assault? I put this plot up here for us to just think about. This was recently put together looking at estuaries all around the U.S. and outside the U.S., looking at eelgrass loss. And the thing that I want to point out to you is that once we get up to nitrogen loads of about 175, we lose everything. All the grass is gone. But for us, to me what's important is when we look down at the low end, why is there such an incredible range? Why at the same level can we have a 5% loss or 100% loss of seagrass at the same nitrogen load? And when you start looking at these estuaries, trying to understand why we see this difference, we find that much of that difference is driven because of the filter feeding organisms that are present in some of those estuaries. What that means is that these filter feeders are basically acting as a weight, that they are buffering this input of nutrient, preventing it to go from total seagrass loss. We spent some time looking at ways that different filter feeders might impact seagrasses. Originally, the first thing that Chris and I had done was looking at hard clams. We put hard clams inside mesocosms, and what we found was that as we increased the density, they would increase or consume the algae, and that would increase light levels, and we'd see a response in the seagrass. And Chuck Wall went, and what he did is he looked at a suite of different ones. Looked at hard clams, oysters, blue mussels, and as he increased the filtration capacity, he saw an increase in light, and that was then an increase in seagrass survival and productivity. I put this up. This is from his dissertation, and it looks at these 3 different species. And the point is, when you look at the turnover times, every one of his mesocosms had a significant increase on seagrass productivity except for one. It was this one. This one failed to have an impact, and the reason was that the turnover time of what was going on in those mesocosms was insufficient to keep the light levels down or up for the seagrass. And so we can take these kinds of data, and now we can ask the question, how much suspension feeding do we need to have an impact on the algal biomass in Shinnecock Bay? This last summer, Chris and his crew were asking the question, not all shellfish are the same. So if we look at certain shellfish, which ones may be better adapted to dealing with the problem that we have in Shinnecock? So he focused on the oyster, the hard clam, and then a gastropod, the slipper shell, which feeds in a slightly different way, which is important for the story. And this summer he had 4 sites that were all, we just draw a line right here. This is where the water quality starts to get bad. So this is on a line of, decreasing water quality, and then his control or outside group over at the marine station. And during this last summer, brown tide existed, and you'll see that most of the sites, all the sites were impacted by brown tide. Quantuck and the Pinnamus Creek were the worst, but the only one that wasn't impacted was the outside outlier group here at the marine station. Not only that, if we look at the temperature of the water, we see that all the sites had temperature for a long period of the summer that was above a temperature that would be stressful to many shellfish. So when we compare the different ability of these different organisms to clear the water, to filter, you can see that to pour in the bad water quality, the slipper shell was the winner, but all of them were not great. There was this really substantial increase by the slipper shell, and the reason that they were able to do this was because these organisms, instead of processing particles over their gill and sorting them out, they use a mucus net. So they're able to get really, really small particles, which gives them the advantage in these areas where they have very, very small phytoplankton, such as the brown tide. But then you'll see as we get away from bad water quality over by the marine station, the oysters were the dominant winner when water quality wasn't terrible. What about survival of these different organisms under the brown tide? You'll see that the clams were the ones that had the worst time. They were the ones who did not deal well being there, and the others did not see a reduction in their survival. And if we look at the growth, one thing that's very apparent from this slide is that the oysters didn't have an impact on their growth rate as a function of whether or not brown tide was present in the water or not, but the hard clams, when the brown tide was there, had significantly reduced growth rates. So the point of that is that we are now looking at 3 different species that are here in Shinnecock. They do their thing differently, and some are going to be better suited for different places if we were going to try to move or enhance densities of these to try to fight against that phytoplankton biomass. Lucas has a poster out there asking the question, can we bring back these resources? So if you're interested in this, be sure to stop by his poster and ask him a question or two about that. I've been talking about shellfish because, again, I'm centric on seagrass and the way that these shellfish could enhance light levels to them. But for a long time I was interested in what impact, how our diversity of the seagrasses themselves. Do we have a lot of diversity, low diversity? We could argue that because we've had the wasting disease in the 30s where we know we lost 80% of our grass around the island. And in the 80s we had brown tide and it suggests that we lost 40% of the grass in our South Shore estuaries. What's left? And that might be really important then on our thoughts of how we restore our situation. So Jamie, one of our students, looked at different populations of seagrass, Fisher's Island, Connick, Great South Bay, Shinnecock. What he found was that certain populations were very similar. In Great South Bay, you couldn't distinguish between east and west. When we compare Great South Bay to Shinnecock, it starts to diverge, but there's still lots of overlap. But when we compare Great South Bay with Fisher's Island or Long Island Sound populations, or Shinnecock with Long Islands, you see that now we've got some significant separation in these populations. It's telling you these are different populations. And so just like we had different shellfish that existed under different situations and thrived, some better than others, the question is, do we have different populations of grass that now are adapted to different lights or different temperatures? And if so, can we then use those seed stocks as what we would use to our efforts to restore seagrass under different environmental conditions? So we're going to be asking those questions this summer in some controlled experiments.